The formation of blood cells is a vital process in the maintenance of the formed elements that can be found in the blood. This process can be referred to as hematopoiesis or hemopoiesis, whichever you prefer to say. Now, the formed elements that can be found in the blood are very, very diverse. We have red blood cells, and we have white blood cells, and we'll also have platelets. So there are three unique groups within the formed elements that can be found in the blood. All of our body's red blood cells and all of our body's platelets are all going to be the same as each other. However, there are many different types of white blood cells, also known as leukocytes, and those are all going to need to form in different ways. Some examples of the white blood cells that we have are eosinophils, neutrophils, lymphocytes, basophils, and monocytes. And each white blood cell is going to play a different role for the blood. So we can see that blood contains lots of diverse types of cells. However, all of the blood cells that I've just listed come from one of the same type of stem cells known as the pluripotent stem cell. The pluripotent stem cell is going to differentiate into one of two cell lineages, the myeloid stem cell lineage or the lymphoid stem cell lineage. And each lineage is going to give rise to various types of cells that are found in the blood. I will show you a diagram including all of these cells on the very next slide. The numbers of each of these cells are going to be very different inside of the blood. Despite the turnover rate of these different types of cells, the red blood cells and the platelets remain rather constant in their concentrations. However, the white blood cells numbers are going to change more variably. Due to the roles of white blood cells, which is going to be protection from foreign pathogens, white blood cells numbers can go either up or down depending on the presence of dangerous pathogens invading our bodies. So we're going to talk about each of these blood cells, but I first want to mention where each of these blood cells come from and discuss a little bit about the lineage of each. So you can see the pluripotent stem cell is the common ancestor to all of these cells, and you can also refer to the pluripotent stem cell as the hemocytoblast. Now there are two major cell lineages that we can see that the pluripotent stem cell will differentiate within. And what's very important is that once the pluripotent stem cell has differentiated into one of these other types of stem cells, it cannot change back. We can see the myeloid lineage over here and the lymphoid stem cell lineage over here. The lymphoid stem cell lineage gives rise to what are known as lymphocytes. These are the T lymphocytes and the B lymphocytes. This lineage contains less cells, and so it tends to be a little easier to remember. But the myeloid lineage, shown over here, is going to contain many other different types of cells, such as the red blood cell, the platelets, as well as the eosinophil, basophil, neutrophil, and the monocyte. All of these cells come from the myeloid stem cell lineage. So as these cells develop, they start to differentiate into different types of cells. There are different characteristics between the myeloid stem cell lineage and different characteristics that can be found in the lymphoid cell lineage. And so these two different cell lineages, as they have different characteristics, are going to develop differently also. Now, one thing that you can see about this line of cells is that each of these cells that can be found here ends in the suffix blast. And you can also see they all contain the same color. So this precursor cells or blast cells get this lightish blue color. A blast cell is a more immature cell that is forming into a more mature version of itself. So if you ever see blast tacked onto the end of a cell's name, you know that it's referring to some type of precursor cell. We'll see within the lymphoid stem cell lineage, we'll have T lymphoblasts that will become our T lymphocytes. Site means cell. Our B lymphoblast is going to eventually become a B lymphocyte. And the NK lymphoblast will become the natural killer cell. So the T lymphocyte is also often referred to as the T cell, and the B lymphocyte is often referred to as the B cell. Naturally, the natural killer cell gives the name the NK cell. So each of these are very nicely named. Now the myeloid stem cell lineage is a little bit larger and contains more cells. So we have the proerythroblast, which is going to eventually become reticulocytes and give rise to our red blood cells. And we often tend to call these erythrocytes. So if I do ever use that terminology, you'll know that I'm referring to a red blood cell. The megakaryoblast is an extremely important cell because it's eventually going to give rise to our platelets. Platelets are cell fragments off of this larger cell known as the megakaryocyte. And so this cell is going to fragment its cytoplasm, and that is what makes up our platelets. We'll find the eosinophilic myeloblast will eventually form into the eosinophil. We'll find the basophilic myeloblast eventually form into the basophil. And we'll find the myeloblast will eventually give rise to our neutrophils. And we're going to go over each and every one of these white blood cells. We'll talk about their different characteristics. But right now, I just want you to start getting an idea of the different progression in the formation of these different cells. We're always going to start with some type of blast cell, and then that's going to take us to each of these more mature cells. Now, I'd like you to know the blast forms of each of these cells, but as we go through this, these names will become a lot easier for you all to memorize. Lastly here we'll have the monoblast, and that will eventually become the monocyte. So that would conclude the myeloid stem cell lineage. Again, I know that there's quite a bit on this slide, but as we start to elaborate on each and every one of these cells, you'll start to be able to differentiate the characteristics of each white blood cell, and you'll start to be able to put a focus upon which cell lineage each of these cells come from. This slide is going to become very important as you study this presentation. There is one more layer of cells. These cells are sort of more miscellaneous than anything else, but we find the plasma cell will eventually come from our B cells, the macrophage will eventually develop from our monocytes, and the mast cell is a very unique cell that does come from the myeloid stem cell lineage. However, it's grouped as a tissue cell. As they sound, tissue cells will make their way into the tissues, and they exist within the tissues. So this slide encompasses the origin and the lineage of all of our blood cells, and they'll all be able to be traced back to the pluripotent stem cell or the hemocytoblast. So first and foremost, we're going to start with the red blood cells. The red blood cells are by far the most common cells that we will have in our blood. In a healthy adult male, we'll find 5.4 million red blood cells per microliter of blood. And in a healthy adult female, we'll find 4.8 million red blood cells per microliter of blood. We will often refer to red blood cells as erythrocytes. So this is another name that I would like you to be very, very comfortable with. This is a very, very common name for red blood cells. Now, the major role of our red blood cells is transport. The red blood cells are the cells that are going to play the major roles in our body for taking gases and other substituents around the body. And the most important thing that our red blood cells are going to carry is oxygen. And this is facilitated specifically by a protein that's found heavily in red blood cells known as hemoglobin. Hemoglobin is a very, very specialized protein that has 4 major subunits, and we can actually see hemoglobin shown here. There's 4 different subunits for each of these. And each subunit of hemoglobin contains what's known as a heme group, which is shown over here. Each heme group contains an iron atom. And the iron atom that's associated with the heme group is what gives blood its very metallic taste. Now, throughout the entirety of all of our exposure to anatomy and physiology up to this point, we've known that somehow oxygen makes its way into our body tissues, but we've never truly discussed how that happens. So what we find is that as we inhale, we bring oxygen in, and these red blood cells contain lots and lots of hemoglobin. And so the oxygen eventually is going to get bound up in the hemoglobin. Each iron-containing heme group is able to bind 1 molecule of oxygen. So therefore, every molecule of hemoglobin is able to carry 4 oxygen molecules. Now, red blood cells are a very, very unique type of cell. They do not have a nucleus, and they really don't contain any other organelles either. Much of the contents inside of our red blood cells is hemoglobin, in fact. So the function of red blood cells is going to be focused upon carrying oxygen. Now we'll see that the shape of the red blood cell that we see here is also going to play a big role in allowing this red blood cell to transport oxygen throughout the body. We describe the shape of red blood cells as a biconcave disc, and you can really see that from this section down here. We have what looks like a circle in this picture, but as we're able to see, the two centers of this disc are eventually impressed towards the center. And the purpose of this is to increase the surface area of the red blood cell to allow for more diffusion of oxygen. The more surface area our red blood cell has, the more space that gas has to diffuse in and out of that cell. The life cycle of our red blood cells is about 3 months or 120 days. There is a steady turnover of red blood cells, and it is the spleen and the liver that are going to filter these red blood cells out after they have been fully used up. Once broken down, we're then going to take the pieces of those red blood cells and reuse them. So we spoke a few slides ago about the pluripotent stem cell. That's going to give rise to the myeloid lineage, and the myeloid lineage will then in turn give rise to our red blood cells. This specific process of making new erythrocytes is what's referred to as erythropoiesis. We will find that the kidneys release a special hormone known as erythropoietin. And erythropoietin is what's going to stimulate the process of erythropoiesis. And we see this negative feedback loop shown here on the right side. So one of the major stimuli that's going to stimulate this feedback loop is hypoxia, which refers to a decreased concentration of oxygen in the bloodstream. So you can see in this example, the controlled condition is oxygen delivery to the tissues. If we do not have enough oxygen delivery, that's considered hypoxia. So some type of stimulus is going to decrease our oxygen levels and give us hypoxia. And that's what's going to be sensed by the kidney cells. The cells in the kidneys are constantly going to be sensing for hypoxia and decreased oxygen levels. And when the kidney recognizes that our oxygen levels have dropped, it's then going to release erythropoietin into the bloodstream, and that's going to make its way into what's known as the red bone marrow. And once erythropoietin has made its way to the red bone marrow, that's going to stimulate proerythroblasts to differentiate into reticulocytes, and then reticulocytes will eventually become our erythrocytes. As we start to make more and more erythrocytes, that's going to increase our oxygen carrying abilities and return our controlled condition back to normal. We can restore the hypoxia that initially started this process. So this is known as erythropoiesis, the formation of erythrocytes, and that is regulated by this hormone erythropoietin. This process does include reticulocytes, which is the precursor cell to red blood cells. The proerythroblast is going to release its nucleus, and that causes our reticulocyte to take the characteristic shape of the biconcave disc. And soon after they're released into the bloodstream, they will become mature red blood cells. Some pathological conditions that relate to our red blood cells are polycythemia and anemia, two opposite sides of the same coin. Polycythemia is essentially referring to levels of red blood cells that are too high, too many erythrocytes that are found in the blood, while anemia, on the other hand, refers to a lower amount of red blood cells that can be found. Now, with fewer formed elements in the bloodstream, anemia is going to result in a more watery type blood, while polycythemia, where there are too many formed elements, is going to increase the viscosity of our blood. And another way to say that would be to increase the thickness of blood. When there is cancer of the bone marrow, which is where our red blood cells come from, that will result in uncontrolled division and then lead to more and more blood cells that can be found in circulation. Now, this would not be a condition we want to be left with. However, a secondary polycythemia is a more normal reaction that can occur when we go to higher altitudes. When we find ourselves in altitudes that are much higher than sea level, we'll see that the oxygen up there is much thinner. There's less of it that can be found at those higher locations. And since there's less oxygen, that can often lead to mild hypoxia, which will eventually lead to more and more red blood cell production. Another related phenomenon is known as artificial polycythemia, which can also be called blood doping. An athlete that's utilizing this procedure would pull blood a few days before whatever their athletic event would be, and on the day of the event, they're going to replace that same blood, and that will result in higher counts of red blood cells, and the more red blood cells we have, the more oxygen can be carried. So we end up with a higher oxygen carrying capacity. But if you are an athlete, I would not bank on this because many committees ban this practice. The next major group of cells that we'll find in the blood are the white blood cells. White blood cells are also commonly referred to as leukocytes, and these are going to be characteristically very, very different than our red blood cells. We're not going to find the same cellular makeup because the white blood cells and the red blood cells play two different roles. We will need a nucleus and cellular organelles to carry out the protective function of the white blood cells. However, we're not going to be transporting anything, so we will not need hemoglobin in our white blood cells. Hemoglobin also, in fact, contains a reddish pigment, and that is what gives blood its reddish color. There are two groups by which we classify our white blood cells, and those are the granulocytes and the agranulocytes. Now we can see that this group says granular and this says agranular, but that's another way we can say that. A granulocyte versus agranulocytes. Granulocytes are named because under the microscope they contain microscopic granules, and agranulocytes do not. Now you may be wondering what a granule is. And a granule is just a pigment under the microscope. You can see that they refer to these vesicles that get stained when put under the microscope. So these cells contain those. This would have been one of the first ways we had to identify cells was based upon the way they looked under the microscope. So the granular leukocytes, or the granulocytes, are the neutrophils, eosinophils, and basophils. And the agranulocytes, which do not contain any cytoplasmic granules, are the lymphocytes and the monocytes. Each one of these white blood cells is going to carry out a unique role in the protection of our body. Now, the white blood cells have a much longer lifespan than our red blood cells have. They're going to patrol the body, searching for different pathogens that have made their way into our body, and their main purpose is to destroy them. Something that many folks do not know about white blood cells is they're not actually contained within the blood alone. They're actually able to escape through the blood vessels and travel throughout the body. This is a process known as diapedesis. You can see that these neutrophils have these special proteins on their outer cell surface, and whenever they're ready to escape the bloodstream, they're able to roll along the inside and eventually squeeze out between two of the endothelial cells. Then these cells would be out of the bloodstream and able to make their way throughout the body to the location of an infection or inflammation. White blood cells use adhesion molecules known as selectins to attach to the endothelial cells and make their way out of the bloodstream. This is an important function of our white blood cells, as many infections occur outside of the bloodstream. These white blood cells need the ability to escape the bloodstream so that they can find these different pathogens that are compromising our body's health. Leukopoiesis is the name of the process to refer to white blood cell formation. The lymphoid cell lineage gives rise to our lymphocytes, while the myeloid stem cell lineage is going to give rise to our neutrophils, monocytes, eosinophils, and basophils. So erythropoiesis was the formation of red blood cells. Leukopoiesis is referring to the production of white blood cells. So erythro is going to refer to our red blood cells, and the prefix leuko is going to indicate that we're talking about our white blood cells. Now, we're definitely going to go over each and every individual white blood cell of importance. But first and foremost, I want to also introduce you to some terms that relate to our white blood cells. So just as we had polycythemia and anemia, we also have the terms leukocytosis and leukopenia. Leukocytosis is a condition which refers to our white blood cells levels being increased. And leukopenia is a condition which is the opposite, that refers to a decreased levels of white blood cells. If there are pathogens that have made their way into one of our body's systems, it's normal for our white blood cell counts to increase as we need to make more white blood cells to fend off those invaders. Leukopenia, on the other hand, is going to refer to an abnormally low level of white blood cells. Radiation and chemotherapeutic agents can often affect our white blood cells levels and cause them to drop significantly. This is a very, very dangerous condition because without white blood cells present, we're not going to have any way to protect against foreign invaders. Leukemia is the name of a condition which involves cancer in the bone marrow. And the result of leukemia is many white blood cells that are released that are not properly able to yet protect against pathogens. Essentially, before these white blood cells are mature, they're released, and they're unable to offer the real protective function that white blood cells are supposed to bring to the table. Lastly is infectious mononucleosis. This is a condition that is caused by the Epstein-Barr virus. One of the most common ways that this virus enters the body is through intimate oral contact, such as kissing, which gives it its name, the kissing disease. You may have heard of infectious mononucleosis before, as it's commonly referred to as mono. The Epstein-Barr virus eventually is going to multiply in the lymphatic tissue and make its way into the B cells, which serve as their primary host. Symptoms of mono often include fatigue, dizziness, headache, sore throat, and a fever. Unfortunately, there's no cure for mono. However, this tends to go away after a few weeks. So as was discussed on the previous slide, leukocytosis is a condition which refers to an increased count of white blood cells. Now this does normally occur when there is some type of infection going on. If some type of bacteria or virus has made their way into the body, we're going to increase our numbers of white blood cells to prevent the actions of that bacteria or that virus. We want to be able to overwhelm the pathogen that's made its way in. So in fact, an elevation in our white blood cell levels can often indicate an infection, and this can be sensed in our blood. Oftentimes, if leukocytosis is indicated, a differential white blood cell count will be ordered by the physician to see specifically what type of conditions can be related to the leukocytosis. A differential white blood cell count just specifically refers to an overall count of each of the 5 blood cells shown below: the neutrophil, eosinophil, basophil, lymphocytes, and monocytes. Both high and low counts of each of these cells can indicate certain conditions and certain issues going on within the body. Since each of these cells carries out a unique role in our body's immune system, increased or decreased levels of any of these 5 cells can indicate certain conditions or problems that would not otherwise be known. For example, neutrophils are very focused on fighting bacteria. They're very good at preventing bacteria and shutting off their toxins. So if we have a high count of neutrophils, this can often indicate that we have a bacterial infection. Another example is the eosinophils. Eosinophils are specifically activated when parasitic worms have entered into our systems. So if we have an increase in the levels of our eosinophils, that can indicate that our body is dealing with a parasitic infection. Page 681 in your textbook shows some conditions that could lead to either a high count or low count of each of these cells. So on the next few slides, we're going to summarize and introduce each of the white blood cells and our red blood cells. So on this slide, you can see that we're going to compare and contrast our red blood cells to our white blood cells. And then following this slide, we're going to speak a little bit about each of our white blood cells, and we're going to start to differentiate them from each other. So first and foremost are the red blood cells. These are known as the erythrocytes, and as we discussed a few slides back, these have the shape of a biconcave disc, and there are no nuclei or organelles that can be found inside of the red blood cell. There is going to be a large amount of hemoglobin, and that's going to be used for oxygen transport in the blood. And you can see that function is listed over here. Remember, hemoglobin contains 4 subunits, each with a heme group that will allow for the transport of 4 oxygen molecules per hemoglobin protein. Now, in terms of the formed elements that we have in our blood, the red blood cells are definitely the most common ones. We see that in females, there's going to be 4.8 million red blood cells per microliter of blood, and 5.4 million red blood cells for every microliter of blood in males. There is a relatively fast turnover of red blood cells, as each red blood cell only exists for about 120 days or 3 months. Now, the white blood cells are much less common in the blood. We see that we only have about 5,000 to 10,000 of these white blood cells per every microliter of blood. And again, this is a range because this is going to constantly be changing. If there's some type of invader that's made their way into our body, those white blood cell numbers are going to go up. As their role is primarily for protection of the body, we want to have more of these so that we can more easily fend off the pathogens that are endangering the body. So let's start to explain the different white blood cells and also differentiate the granulocytes from the agranulocytes. So first and foremost, we're going to go over our granular leukocytes. There are 3 of these: the neutrophil, the eosinophil, and the basophil. These are called granulocytes because they contain cytoplasmic granules, and you'll see that on the pictures that are associated with each slide. So first and foremost is the neutrophil. The neutrophil is the most common of all white blood cells. You can see that the neutrophils make up almost 3/4 of all of our white blood cells. Now specifically, if you look at this picture, you can see these little dots. These are the cytoplasmic granules, and again, that's what gave them their name. Now neutrophils tend to be easy to spot because they have lots and lots of lobes to their nuclei. You can see their nucleus isn't a typical spherical shape like we're used to seeing. There's different lobes that can be found as part of the neutrophil's nucleus. So oftentimes we'll see that they have between 2 and 5 lobes. But if I do decide to ask you about a particular amount of lobes on the test for neutrophils, I will use between 3 and 5 lobes for the neutrophil, since many of the other blood cells will have 2 lobes. So it is the multiple lobes of our neutrophil that allows us to identify this under the microscope. Now neutrophils specifically are going to be mostly focused on bacteria. There's many different types of pathogens that make their way into the body, and different white blood cells are going to specialize for each. And neutrophils are our main player for bacteria. So they're going to synthesize this enzyme known as lysozyme. Lysozyme is an enzyme that's going to lyse the cell membrane of different bacteria. They're also going to use oxidants such as superoxide anion and hydrogen peroxide that will also be deadly when projected at bacteria. Now the eosinophils are going to be the second most common of our granulocytes. These make up about 2 to 4% of our white blood cells. Eosinophils are very characteristic to look at because their nuclei tends to have 2 lobes, and we'll find these very, very reddish staining granules. You can see this color is much different than the neutrophil's color was. These red or orange staining granules are going to indicate we're looking at this particular cell. Now, while eosinophils do several things, one of their main actions that I want you to focus on are parasites. Eosinophils are going to be utilized when parasitic worms especially enter the body. So if we have an increased level of eosinophils, that's going to indicate we might have a parasitic infection. Our least common granulocyte is the basophil, and we could also say that the basophil is the least common of all white blood cells. These make up only up to 1% of all of our white blood cells. Now we will see 2 lobes to the nuclei of the basophils. However, as you can see, these basophils are very, very darkly staining with lots and lots of cytoplasmic granules. When something stains darkly under the microscope, we say that it's a basophilic dye. And that's why this cell got the name of the basophil, because there's going to be these very, very dark bluish purple granules that are obscuring the view of the nuclei. When you see basophils, you want to start thinking about allergic reactions and the inflammatory response. Basophils are going to release a chemical known as histamine, which is going to be a major mediator in the allergic reaction. Histamine is also the target of a class of drugs known as antihistamines, which will suppress the allergic reaction. So you should associate the term basophil with allergic reactions. Now, as for the agranulocytes, these are not going to have cytoplasmic granules. That's how they got their name. And the more common of the 2 types of agranulocytes is the lymphocyte. Now, the lymphocyte is an extremely important white blood cell. And these are also going to be considered the second most common white blood cell that we have. These make up about 1/5 to 1/4 of all of the white blood cells that can be found floating around in the blood. Now, there are several types of lymphocytes: the T cells, the B cells, and the natural killer cells, and we'll elaborate a bit on each of these. But in terms of identification, all of the lymphocytes look relatively the same, and they have a very characteristic appearance as well. So the nuclei of our lymphocyte is much more rounded than that of any of the granulocytes. We'll have a much more rounded nucleus, as well as a rim of cytoplasm that you can find around the outside of the nucleus. Now, as we start to define the functions of our lymphocytes, we will have to distinguish between the T cells, the B cells, and the natural killer cells. Now, we're going to discuss each of these cells a bit more when we get to the immune system, as that's where their primary roles are going to fall within. However, what we need to know for now is that B cells are going to eventually control our body's humoral immunity, which can also be referred to as antibody-mediated immunity. B cells are the specialized cells that make antibodies and release them that will ultimately stop pathogens. The action of T cells, on the other hand, tends to be a little bit more hands-on. There are several types of T cells, and their major role is going to be to stop viruses and cancer cells that have entered or formed. And lastly, as their name suggests, the natural killer cells are specifically going to focus upon infectious microbes. Now, the last of our agranulocytes and the last of our white blood cells is the monocyte. The monocyte's appearance is very characteristic, as it has this indentation in the nucleus. In fact, this cell is going to look a lot like a lymphocyte does. However, the nucleus tends to be kidney bean shaped or horseshoe shaped. And that is what's going to distinguish our monocyte from a lymphocyte when looking under the microscope. Lymphocytes also tend to be the smallest leukocytes, while monocytes are going to be the largest leukocytes. Monocytes are considered phagocytes, which means that they carry out the process of phagocytosis, which is where one of our body cells is going to find a pathogen and completely devour and consume that dangerous cell. These are going to be the third most common, as they make up between 3 and 8% of our white blood cells. So that encompasses all of the white blood cells that we're really going to mention in this presentation, as well as the red blood cells. However, there is one other formed element that can be found in the blood, and those are the platelets, which can also be referred to as thrombocytes. The main role of platelets in the blood is clotting. We want to be able to prevent the profuse loss of blood, and platelets are just the players that do this for us. Platelets are little cytoplasmic fragments of the cell known as the megakaryocyte. Platelets will circulate in the bloodstream until some damage has occurred to one of the vessels. When a blood vessel has ruptured or broken open, that's going to release certain factors that will stimulate platelets to start the clotting cascade. Platelets are the second most common factor that can be found inside of the bloodstream. You can find between 150,000 to 400,000 platelets per microliter of blood. So from most common to least common, we'll find our red blood cells, then our platelets, and then we'll find our white blood cells. And if we want to rank our white blood cells, we'll find our neutrophils as the most common white blood cell, then our lymphocyte is going to be most common after that, and the remaining three will be the monocytes, the eosinophils, and the basophils in that order. So platelets are vital for what's known as the blood clotting cascade, and we can also call that process known as hemostasis. Hemostasis is such an important process. As you all know, our bodies are not indestructible, and there are different stimuli that lead to breakage in blood vessels or damage to the cardiovascular system. Anytime we bleed and release blood, that's going to indicate that some type of blood vessel has been opened up and blood is escaping from the cardiovascular system. Now, if we didn't have platelets, this process would continue indefinitely. So platelets are a very important player in stimulating the clotting of blood. We will discuss the process of hemostasis next.